Hello, I'm glad to have you joining me. So we will be covering this chapter in a bit faster pace than the earlier chapters. And this is because a lot of this material is simply expanding and giving applications to what you have already learned about genetics. We remember from our earlier chapters that a gene was a sequence of DNA made of base pairs that codes for a product. And most often, this product would be a protein of some kind or result in a certain trait that we see in an individual. So, what is a mutation then? Well, a mutation is a change in this base sequence that codes for a gene. And let me get one misconception out of the way immediately. Actually, mutations are not that rare. By this I mean that it is not uncommon for small changes to happen in the bases, say a difference of one base. In fact, most of the time that change will not result in a significant consequence, so it goes unnoticed. And there are many causes why a mutation may occur. These include an error in the copying process. Remember all those checkpoints, so these are not very common. We also have bits of DNA jumping around to places where they are not supposed to be. And finally, it is also possible that we get mutations due to the environment or chemical effects, or something may go wrong with the repair process. And here we have one example of a gene mutation. And this would be due to a transposon. And from our previous slide we remember that it refers to a bit of DNA jumping about. And this can cause a change, for example, in the color of the corn pigment. And this brings me to a great place to mention something. In fact, what you see as this yellow corn cobs that we all know, that is not what original corn looked like. That looks like it does just because we have been selectively breeding to make it look like that over many generations. So this would be genetic engineering that farmers do all the time. Instead, I have here a picture of native corn that we grow on our ranch. Isn't that pretty? And that is what the corn should look like. It may be that it has a higher nutritional value too. Well, I just wanted to share this picture as I always get super excited when it is the corn harvest time, as you never know what colors you will get until you'll peel back the husks. It's like little jewelry pieces, I think. In our ranch, we try to preserve the native species and run it in a sustainable and ecological manner. And one of the things that many visitors often find surprising is that this thing with the color of the corns. So, mutations can be of different kinds. And there are two that I want us to discuss about, just briefly. So, point mutations are mutations that take place at a single point. And this is single small change can either have minor or major consequences. And one example of this that we have seen earlier was the sickle cell disease. And what happened there was that a single base was different, and that caused the single amino acid added to the protein to be different, and that in turn changed the entire shape of a red blood cell. So this would be an example of major consequence of a single point mutation. The other type of mutation that I wanted to mention was the frameshift mutation. And here we have an extra or a missing nucleotide. And that causes usually much more severe consequences. This is because all of the amino acids coded by these groups of 3 nucleotides are affected. This is because taking away or adding one kind of moves all others to a wrong place. So, that is our introduction to the genes and gene mutations. Let's look at chromosomal mutations next.